Hello everyone, welcome to today's live broadcast, More from Less, high quality data from low quality samples. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Perkin-Elmer. I am Judy O'Rourke of LabRoots and I'll be your moderator for today's event. We have a few important announcements before we begin. This webcast is designed to be interactive and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Andrew Barry, Director of Product Development and Marketing for the Broad Institute Genomics Platform. I will now turn it over to Andrew Barry for his presentation.
Thanks, Judy, and thanks to all for joining today. So as Judy mentioned, I'm responsible for product development within the Broad Institute Genomics Platform, and really our goal is to provide our internal as well as our external community with a set of products and resources to produce high-quality data. And as we get into the different breadth of applications for which genomics is being used, we really need to expand the sort of threshold for which we can accept samples, and a lot of the work that's gone on within the Broad Institute Genomics Platform has really been focused on developing strategies for making the most out of the samples that we received, and developing strategies whether within a project or across technology that really allow us to produce the highest quality data we can regardless of what we get as the input to the process. So I'll be speaking a little bit today about some of the different strategies that we've employed. Just moving on to the next slide, I'll give you a little bit of a background on the Broad Institute itself. For those that might not know, the Broad Institute is a nonprofit biomedical research enterprise that was founded in 2004 by MIT, Harvard, and its affiliated hospitals, the Whitehead Institute, and our founders, the philanthropists Eli and Edith Broad. And the goal is really to accelerate the pace at which the world conquers disease. We're located in Boston, Cambridge actually, one of the world's most influential crossroads of academia, industry, and medicine, and we're bringing together leading biomedical scientists from our community, again, with the goal to develop bold new ideas that address the most urgent and ambitious problems that the world is trying to solve related to human health. Really at the core of this is a collaborative model. This is between our own researchers and the hospitals that are within our community, external groups, government agencies, philanthropic donors, and really, again, the goal is to create the necessary models to take on some of these ambitious challenges, and we do that with a very deeply collaborative spirit. We collaborate as broadly as we can, and one of our goals is to share our methods, resources, and data with the ultimate goal of enabling others to accelerate discovery. And we're constantly looking for new ways in which to disseminate the learning and output of our activities and get the information that we're learning, whether it be data or methods, or really exposing some of our capabilities externally so that others can leverage them with the external community, and trying to use this as a means to really help the industry overall and anybody that's interested in advancing human health. So the genomics platform itself, it's a fairly big infrastructure, and our goal is to provide high-quality genomic data to the scientific community, both internal as well as external, and really to facilitate some meaningful conversations with the folks that are looking to leverage these data. And the goal there is to ensure that we're continuing to develop the right tools based on real-world problems that researchers are starting to use genomics to solve. And I think that's really speaks to the need for producing as high-quality data out of low-quality samples as we can, because we don't always have the luxury of using our high-quality and high-performing control samples. When things get turned on in a production environment, we typically learn a lot more from the lower-quality samples than we do our what we would use to typically develop a process. We pride ourselves on the scale, quality, and speed at which we can take on some of these ambitious projects, and we're really anxious to see what types of questions folks have as we sort of expand our horizons and take on more in terms of what we're actually able to interface with the outside world with. So, I think part of our culture here in the genomics platform is we've really merged together sort of industrial best practices and applied those to genomics. And we work closely with the MIT Sloan School of Management and benchmark ourselves against, a lot of external industrial best practices and groups that have applied, things such as modular automation, process design, work design, these sort of aspects being applied in a life sciences genomics laboratory is sort of a novel concept, and it's something we pride ourselves on. And really the goal is to create a situation where we can take existing technologies and processes and perform some optimization. And the way that we do that is we typically will take a process and break it down to its individual components and look at those individual process steps and define what the inputs and outputs of those are. Once we do that, we can actually start to optimize within those process steps, and then as we put these things together, what we find is that we're able to develop a level of process control that really helps us to trust the processes that we're running in terms of taking in raw sample and turning that into genomic data. This is just an example that sort of highlights this, where starting with a process where we were running something manually, and on the y-axis that's just a percentage of unclassified reads, which is just a single proxy that we might use to optimize something. But once we start to measure those things and apply some of these techniques and really do that sort of modular optimization within these process steps, what we find is that we're able to generate really higher quality data. And we do this through a lot of different tools. So one is really end-to-end sample tracking, so making sure that samples that enter our process, we have positive sample tracking as they move through. And that's something where, in this case, we're showing a custom consumable that we would use to take in samples and be able to apply that with a barcode and make sure that we have tracking through our LIMS. So at the end of the day, when the data gets produced, it can be associated with the correct sample information that we've received. Building error proofing into the process. This is a concept. Poka-yoke is the technical term for it, but really it's error proofing and creating multiple checks and balances throughout the process that allow us to make sure that the right information is being asked for and being entered into fields that will later allow us to go back and do some troubleshooting should we need to. And having this type of approach to life sciences, again, it's something sort of novel, but really ends up having a pretty big impact. We think about lab automation. It's very integral to what we do here, and we believe it's really a huge part of what we would use to be able to do things at scale, at high quality. We've taken a modular approach to this, where rather than having a lot of different things being done on a single deck with a lot of different inputs and outputs, we tend to break things down into their fundamental processes and have a single piece of automation doing a rather simple task, but doing that highly repetitively. And we found that we can generate some high scales with that. And then, once we get all of this information that we're collecting about our process back, building some of these dashboards that allow us to monitor our processes and make sure that they're actually in control. And when they're not, be able to dig in deeper and find out what the root cause of some of those issues are. So it's really a combination of the lab processes, being able to capture the information up front, being able to take those and embed them in a data warehouse, and then at the end of the day, associate those with the sequencing metrics that might be associated, pull those things back together, and develop some real-time analytical tools. And what we find is that when we apply these things, and this is just a specific example, you can see that all of a sudden that percentage of unclassified reads goes down dramatically and is a process that's in much higher control. So these are the types of things that we would typically apply to any type of sample type, but when we start to look at more challenging sample types, typically they force us to think outside the box a bit and start to apply some of these techniques in a slightly different way that is more specific to different, more challenging sample types. Just to give you some information on the platform and some of the scale at which we're operating. So we currently operate about 40 HiSeqs, and those are split between 2000s and 2500s. We have 10 MiSeqs, a single NextSeq 500, and we have 14 HiSeq X's or 1.4x 10s, however folks typically think about that. These are just some of the numbers in terms of the sort of throughput, in terms of, this is from 2009 to 2014. I haven't updated this with 2015 numbers, but, over 120,000 exomes. RNA-seq is really upticked in the last several years. We're producing more human genomes now than ever before at a rate of about somewhere between 1,500 and 2,000 genomes per month. In fact, in the month of October of 2014, we actually produced a single human genome every 32 minutes using the HiSeq-X's. So those were being run at full capacity, and that's always our goal, is to run those at as high capacity as possible. And fortunately, we've been able to achieve that over the past several months. We do a ton of genotyping as well. In terms of samples stored, we're collecting samples from all over the world in different formats. So we have about 1.2 million samples stored, with the capacity to store about another 2.5 to 3 million on premises here. And then, just looking at the total output, that's 3,000 terabases, so that's 3 times 10 to the 15th bases. So quite a large scale, and obviously a lot of this is enabled by the advances in the sequencing technology itself, and we've continued to sort of build the infrastructure around those sequencers to be able to do this at scale and to do it reliably. So when we think about how we interact with the outside world, really we think about, we have Broad researchers that are sort of PIs, and the way that the Broad is sort of organized is between what we call scientific programs and scientific platforms. So the right way to think about a program is that these are really the high-level scientific questions that are being asked, and the platforms themselves are really the nuts and bolts and the factories that live within in different application and technology areas that allow us to look across those and use whatever tools we need to apply to be able to answer the right question. So that's the Broad Institute Genomics Platform. Broad Institute Genomics Services is really our way of interfacing this with the outside world. So this is really our way of taking the things that are our core capabilities and making those available to the external world. So we do that through Broad Institute Genomics Services. You can visit our website at genomics.broadinstitute.org. And really, when we think about that, it's everything from really transactional fee-for-service products that you can browse on our website and understand the inputs and outputs of. And then there's really more custom solutions where we're taking different components of this and putting them together to really address a unique problem that might be specific to a disease area or a sample cohort that one is trying to study. And then in addition, there's situations where maybe the technologies that exist today don't quite give us what we need to answer those questions, and that's where we engage externally with groups to build more deep scientific collaborations and really build something that's totally new to the world. So today, we'll talk a little bit about sort of the breadth of the applications for which NGS data is used and understand some of the sample-specific challenges in terms of maximizing data quality for those. And then we're going to discuss some of the strategies that we've employed here to overcome sample quality with the goal of improving the data quality that comes out of the sequencers at the end of the day. So, really when we think about this, we've broken this down into sort of 4 discrete areas. One is really the nucleic acid extraction, so how we can take whatever the primary material is and generate as high-quality nucleic acid as we can. The next is really the QC of that. So how do we look at these samples and, number one, look at an assay that's going to give us some information that's going to tell us, the quality and the inherent quality and some of those intrinsic characteristics of the nucleic acid, and then really associate that downstream with what the impact of that is on sequencing data. And what that allows us to do is work with our collaborators and PIs and, try to understand what the path forward is for some of those samples that, maybe are on the edge and maybe we don't want to pass on and spend more money to actually turn that into genomic data. And somebody described this to me as the watchful older sister that's asking, do you really want to date that guy, because the sample looks a little bit sketchy. So that's sort of how we think about some of those QCs. And really a lot of focus here has been on the sample preparation strategies and how we take things from purified nucleic acid and make sure that we can deal with lower amounts but also lower quality nucleic acid and still make sure we can get the data through that. So we'll explore some of those. And then some of the sort of analytical approaches to being able to compensate for what might be lower quality data. And we'll discuss some of the tools that we use to help filter out some of the information that might not be helpful in terms of discovering new variants, some of the artifacts, and then also how we can actually use some of the breadth of what we have in our own experience to be able to compensate for some of the lower quality data. So when we think about what our core product offerings are here, really we think about this as human whole genome sequencing, human whole exome sequencing, human whole transcriptome sequencing, or RNA-seq, and we also have a CLIA CAP-accredited lab where we offer a technical exome with either germline or somatic analysis. And it's important to know that for all of these, or several of these are FFPE-compatible technologies. A lot of the work that we're doing here is focused on oncology, so formalin fixed paraffin embedded samples are a very normal thing for us to process here. So a lot of work has gone into FFPE, but also some of the other more challenging sample types that we'll discuss. So when we think about what these challenging sample types, we mentioned FFPE and what some of the challenges there are. So FFPE tends to be degraded. We have some cross-linking happening there, some deamination events that might take place, oxidation based off of that, as well as challenges with low allelic fraction, and we'll discuss a little bit what the impact of the allelic fraction will actually have on the quality of the data and the sensitivity that we're able to achieve. Saliva is particularly challenging when we do genome sequencing from saliva. We typically will see high levels or varying levels of bacterial contamination that can sometimes be challenging to actually meet the desired coverage to be able to use in some of the downstream analyses. Another example would be blood, where sometimes we see tumor in the normal if we're doing tumor-normal pairing. Oftentimes blood is the normal, and sometimes what we see is that we'll actually have some of the tumor, especially if it's a blood-based ailment, that we actually have a difficult time because there's actually tumor in the normal, so it's harder to use for somatic analyses. When we think about understanding and resolving some of these more subtle artifacts in the data, one of the things that we've done a lot of work with is with FFPE. So FFPE, as we discussed, will introduce lots of different types of DNA damage. Deamination is one of those. So this is where basically due to an oxidation event, we'll convert cytosine to uracil, and that's going to lead to, when we actually do PCR, we'll actually see that we'll have missing incorporations. And what we've done is taken a step back and built some tools that actually flag some of these artifacts as artifacts, and that really is going to help us sort of differentiate the artifacts from a true-false positive or an actual variant. And those are actually part of our standard outputs now, where part of the Picard pipeline actually will develop some of these metrics and actually output them. And it's really important for us because when we're introducing a new process, now we have another thing that we want to make sure works, and we're always looking at some of these values and making sure that they're not creeping up, because they're the type of thing that can creep in. One of the prime examples of this was actually in our DNA shearing, which I'll get into in a few minutes, but we actually saw this start to creep up, and we're able to overcome it fairly easily, but it's one of those things where if you're not looking for it, is something that's going to creep in and can really cause some confusion at the end of a study, where all of a sudden you're trying to whittle down to the real causal variants, and now you have this list of things that are really artifacts that are going to be prohibited from drawing more logical conclusions. We talked about the impact of allele fraction on variant detection. So, really when we think about allele fraction, it's the fraction of DNA molecules that come from a locus that carry a mutation. So when we're dealing with FFP, obviously what we're trying to accomplish is, getting the most cells in there that are actually, when we're doing tumor-normal pairing for somatic analysis, we're actually, really interested in getting the majority of those cells that are going to have the mutation present within the copies that we're putting into the downstream analysis. And that figure on the left, which will show you the output after some optimization, but you can see as we go from, for instance, from the yellow line to the red line, on the y-axis there is the actual sensitivity to detect single nucleotide variants. And what we see is that as we drop in that allelic fraction, you really start to get to the point where it's going to become very difficult. And there's some things that we'll discuss that we can do to overcome this, but it's important to understand, and I think there's a need for newer technologies to be developed to really make sure that we're doing a good job up front between pathology and nucleic acid extraction to make sure that we're getting the highest percentage of tumorigenic cells into our prep. But we'll talk about some strategies that we've employed to overcome those as well. we mentioned the impact of bacterial contamination in saliva samples. We see this typically on a collection-to-collection basis. This is worse when we see things from a more consumer genomics approach because proper protocols aren't necessarily being adhered to, and what basically this is bacteria from the mouth that actually ends up into the extraction and downstream sequencing. So you might have a situation where you have somewhere between 40 and 60% of the reads are actually going to be derived from bacteria, which if you're trying to hit 30x mean coverage on your genome, it makes things fairly difficult to achieve because you're going to have to keep sequencing and sometimes may never get there just based on the inherent contamination that takes place with these events. So now we'll shift gears a little bit and start to talk through some of the various strategies that we've employed here to overcome low quality samples. So starting with nucleic acid extraction, we've done a ton of optimization around the different nucleic acid extraction chemistries, and we mentioned kind of the process where we take things and break them down to their fundamental parts. One of the technologies that we've employed here for DNA extractions uses the chemogen technology. This is a magnetic bead-based approach where we see advantages because the chemogen technology itself it's an electromagnetic rod as opposed to pipetting, so we're able to maintain some of the integrity. Typically when we pipette things through some of the narrow bore pipette tips, what you actually are inducing there is our shearing mechanisms for the nucleic acid. So what we found is that by using the chemogen technology, we're able to get higher quality nucleic acid out of that, and we've applied that to a very challenging sample type, which is dry blood spots, and can now reliably get enough material out of our dry blood spots to go into some of our downstream applications. So, seeing this list just kind of gives you an idea of the breadth of applications and the different types of, primary sample materials that we're able to generate high quality data of from, based on the input that's provided to us. We also have developed a fairly comprehensive suite of RNA extraction products, as well as a series of extraction products that will actually take the same sample and produce both DNA and RNA. So we'll end up with enough material from a single, section of FFPE or a single group of sections from a single block of FFPE that aren't mutually exclusive in the sense that, my DNA isn't going to be exclusive. My DNA quantity isn't going to be exclusive of the RNA quantity. So we're able to generate, both high-quality DNA and high-quality RNA at the same time from the same sample, which is really beneficial. And we'll talk about some applications where we'll start with the same sample and put that into downstream both DNA sequencing and RNA sequencing and actually can draw some pretty cool conclusions out at the end of the day. So moving on to nucleic acid quality control, this is where we've spent a lot of time really building out assays and working with folks in the industry to build out some of these assays that'll allow us to really assess some of the intrinsic characteristics of the nucleic acid. So we're looking at things here like the concentration, the size, the degradation that might take place there, as well as really understanding how much goes into those downstream processes and making sure that there's the amount that you're putting into those is normalized. And what we find is that by putting in the same amount of nucleic acid into some of these processes, you're able to get more reproducible results out on the back end. So one of the most useful assays that we use here is it's a qPCR assessment of degradation. We use a kit from Kappa Biosystems. And basically the way this works is we take 3 different size amplicons and perform qPCR across those. And what we have worked with them to develop is this concept of a Q-score. So what the Q-score does is it looks at the different ratios and abundance of the different amplicons that we've created. And what we can do is sort of look at that and tell how much degradation is actually there in the sample, how much material is actually capable of being PCR amplified. And what that allows us to do is have a prediction of how well this thing is going to actually produce high quality data, and that's what you see in this bottom right figure here, where we actually can predict the probability of success based on this Kappa QC score, where certain samples we'll just not attempt because we know that it's not going to produce enough high quality data to do anything useful with. So again, this is something that we often have an interaction with our collaborators or customers on, and are able to feed that information back and allow them to say, either go ahead with the sample and, I understand I'm not going to get high enough data quality out of that, or, really give them confidence that their sample cohort is going to produce high quality data out at the end of the day. And we can see the impact that this has on the output. So on the x-axis here, what you see is these are all different types of FFPE samples that we've put into this process. And similar to when we discussed the saliva and bacterial contamination, these do tend to vary based off of the actual collection itself. And what we found is that the actual quality that we're able to get out at the end of the day is actually less to do with anything related to the age of the FFP block that we're extracting nucleic acid and sequencing from, but more to do on a collection-to-collection basis. So, just something that we've observed, and you can see that, after we implemented that QC, we're able to generate much higher quality data with the sample cohorts that we're putting through. The other assays that we'll run actually use the PerkinElmer LabChip GX technology. So this is where we'll actually take a look at the size of the nucleic acid and actually look at this genomic quality score, which is a single metric that's attributed to nucleic acid. So this is an electrophoretically separated assay where we're taking the DNA, separating it, and then actually looking at the size that's being achieved there. And developing this Q score allows us to boil that down. So what you see on the bottom there is actually an experiment where there was a degradation of nucleic acid, and we're able to see the corresponding drop in the Q scores associated with that. So that's something that we'll use oftentimes if we really are suspicious about a sample and want to know, exactly how degraded it is before we put it into any of the downstream processes. Similarly, for RNA, I think the RIN is a typical assay that folks will use for general RNA quality. The PerkinElmer GX is what we use here that has an analog RQS, or an RNA quality score, that's roughly the same calculation that would take place in the RIN. What we find is that for more challenging, in particular FFPE samples, this new score, this DV200, is actually a more accurate predictor of downstream success. So what we're looking at is what is the population of fragments that are greater than 200 base pairs, and we can actually calculate that based on the electrophoregram of separating the RNA out in the electrophoretic capillary. So, really, we do a smear analysis, and we're able to output that. And, again, we're using this to predict what will happen. And some of the techniques that we're employing to do RNA sequencing from FFP material really rely heavily on this as a gauge to make sure that we're going to, again, have a good predictor of success downstream. Again, this assay is super easy to use, and we can do this at high scale, and it's to the point where, we can just click a button and output a single metric that's going to allow us to use as a hard and fast cutoff. So we usually set the cutoff at about 30, and down in the bottom right, you can actually see what the correlation is between that DV200 score and the actual yield that we're going to get out of our library prep. And that also correlates to something that we always look at, which is the % duplication. Typically, when we do PCR, what we find is that if we have low input or degraded samples going into PCR, we'll get higher rates of duplication happening. So if we look at the DV200 score relative to % duplication, we typically see those two things inversely correlated, where the higher the DV200, the better the better our duplication is in terms of lower duplication values. So shifting gears to sample preparation protocols, so this is really where we're talking about taking the output of our nucleic acid extraction with these more challenging materials and putting them into some downstream processes that are going to get them ready for sequencing. So that, really when you look at our product input requirements, we're trying to minimize these, and I'll just, mention that the minimum required DNA amount that you see there, that's actually 1.5 times what we actually will use. So we like to give ourselves 2 attempts just in case something goes wrong. So these are the amounts of nucleic acid that we actually will ask for in terms of guaranteeing that the results will be good based off of this. So if we take the samples and actually assay them and generate this material, we can guarantee that the data will actually produce the specified deliverable for a given product. So, you see that those are relatively low across, 250 nanograms, so that means that we're actually taking 100 nanograms into the downstream process. The sort of one outlier here is PCR-free human whole genome sequencing. We ask for 1.2 micrograms. That's actually a 500 nanogram input, and we're working hard to reduce that because we're really happy with the performance of PCR-free human whole genomes as our collaborators, due to the cleanliness of the data in terms of variant calling, as well as, eliminating any issues that we have with base-specific bias. So one of the techniques that we've employed here is really do with the fundamental library prep, and this paper was published, I guess, about 3 years ago, but still gets used routinely here. What you see on the right there is a figure that sort of describes the innovation that we applied here. So this is where, we're doing purification cleanups mid-process, so in between some of the enzymatic steps. Typically what we'll do is a bead-based purification. We use a spry technology where basically we bind nucleic acids to these iron core carboxylated beads, and we're able to do a couple things there. We're able to concentrate or adjust the concentration of samples as they navigate. We obviously want to purify away any active enzymes or unincorporated things like oligonucleotides, and then the other thing that we're able to do with this is an actual size-based cutoff. So we're able to actually, based on the concentration of some of the buffers, and we use a polyethylene glycol buffer there, and based on the salt concentration and the concentration of the peg that's in the buffer, we're able to adjust the molecular weight of the fragments that get carried forward in the reaction. So the novel innovation here was, typically these things happen serially, and there's a lot of steps that take place where there's actually result in a lot of sample transfers between different lab receptacles. What we've done is actually eliminate those transfers. This involves actually leaving the beads in the well during some of the enzymatic reactions. They don't inhibit the reactions. We've done a lot of work to make sure that's true, and they give you the added benefit because every time you're going to pipette things out of one receptacle, whether it's a test tube or a 96-well plate, you're going to leave behind some material there. But so just by leaving them the beads in the well and being able to do your reactions where you're based on the buffer concentration, you're either binding to the beads or eluting off of them, you're able to do it all within the same well, and that allows us to do really the entire sample prep protocol on a single well, which again reduces those transfers and results in much higher yield. So even if we're starting with lower input, we're able to carry more of that material forward downstream in the process, and that's what allows us to take in some of these lower quality samples and still generate high quality data. We talked a little bit about some of these subtle artifacts in our data. So this is a specific example that we were looking into, and this is really this oxidation that was taking place during mechanical shearing. So this was on the Covaris instrument. And what we were able to do is actually look at this, and there's a publication based off of this that you see down the bottom there. But really what this was we're introducing these artifacts into our sequencing data, and we found out it was basically because of the buffer that we're using to shear in. It was, again, a very easy thing to overcome by adding EDTA, which is the chelating agent, into there. So that was able to prevent the oxidation from happening, as it was able to bind some of those metal ions. But it's one of those things that if you're not looking for, can really creep into your data, and it's something that again now we look for across everything. So right now, if we're looking, for example, to introduce a new deparaffinization solution for FFPE, this is something that we want to make sure isn't inducing any of these types of artifacts, because we've seen it in various buffers that we use for some of the exome capture protocols, and have done a lot of work to optimize to make sure that we're not introducing some of these artifacts moving forward. And you can see the impact that we have on the sequencing results. So this Oxidation Q score, again, this is something that's part of the standard output of the Picard pipeline, but something that we'll track and make sure that we're mitigating any impact that might happen. The impact, whole exome sequencing has for a long time been really the bread and butter of a lot of the sample processing here. We developed, really the first scalable method for this, and working with Agile and SureSelect to develop that chemistry, but also automating it and doing it at scale. I mentioned earlier, but we've done over 120,000 exomes here. That's been cited in over 3,800 publications. And you can see the impact that has here, and I think, specifically when we look at some of these things, one of the things that is really impactful. So, you know, in January 2011, we would ask for 500 nanograms. Now we can actually look at this, and that's more like 50 nanograms input. And we're always asking for more than we actually will take, but we know we can push the limits down and still produce high-quality data while doing things like increasing the scale and reducing the failure rates that are associated with those. One of the other things that we've really done here is you know, we've introduced a new design for our exome, and you know, it's if you aren't targeting some of the bases that are really interesting to you, you can't expect to get high-quality data from those. So we've done a lot of work to really optimize the bait design for exome sequencing to make sure that we're getting the best coverage that we can. So we recently introduced and validated the Illumina custom exome, is what we call it, or ICE. So it's a new design for us. It's using the Illumina chemistry, so it's 2 subsequent hybridization and captures, which provides us with very clean data. It's a 38 megabase bait set, and we've done a lot of work to really optimize that in terms of, you know, covering some of these more challenging regions that we've learned from our past experience. One of those things that we've done to optimize that is improving the coverage uniformity across targets with different GC contents. So what you see here on the left is looking at 2 different, our old exome and our new exome, and what we see is that, you know, for things that are higher GC, medium GC, and low GC, we're actually able to you know, on the left side of that red dotted line there, for the high GC, we're able to have far less of those things showing up with very low coverage. And what that means is that, you know, if we have challenging samples, we have a better chance of actually being able to generate high-quality data because we're actually baiting and targeting those regions. You can see this is just an IGZ view of a particularly difficult target. This is ApoE, but we've gone back in and adjusted the bait design to really make sure that we're getting adequate coverage. So, you know, the new, you can see we have more than adequate coverage across that, where the old we were missing that altogether. Not altogether, but when we think about the distribution of depth, this is an analysis that we performed where, you know, we're comparing the average depth of coverage, and what you can see on the right there is that with the new, we're getting a much higher percentage of our targets, I'm sorry, a much lower percentage of our targets under where we feel like our cutoff should be to be able to call the right types of variants that we're looking to use downstream. So, you know, overall, it's a better performing bait. And the impact that we see there is, you know, this is going back to the figure that I had shown at the beginning of the presentation, but it really, the impact on the sensitivity that we're able to achieve is really where, you know, sort of the rubber hits the road, in the sense that, you know, again, these colors will correspond to the allele fraction going into this, so the percentage of the cells that are actually inherently mutagenic, that are actually holding the variant. So again, you know, we're looking at specifically between this red and yellow and the old exome design versus the new exome design. By having better coverage and more even coverage, we're able to, you know, again, with the same amount of data, have a higher degree of sensitivity for things like single nucleotide variants. This is an example where we're actually looking at some whole exome sequencing and whole genome sequencing data from FFPE, and we're actually looking at, you know, how many variants we can call between known and false positive. So we're able to see that, you know, with whole exome sequencing, while we're not getting quite the number of variants that we'll get with whole genome sequencing, we're able to do pretty good considering we're only targeting, you know, 1 to 2% of the genome. So, you know, really just a nice demonstration to show that we're able to generate, you know, relatively high quality data from FFPE, and really the FFPE is not a limitation in terms of the ability to produce that quality. Copy number variance is something that we're doing a lot of work with currently to sort of optimize and be able to do as reliably as possible. This is showing a canonical MDM2 amplification in whole exome sequencing and whole genome sequencing. And, you know, in whole genome sequencing, you can see that we're able to find this much better, but it's still there in the exome sequencing. And again, this is, you know, something that's really hard to do, which is called copy number aberrations from whole exome data from FFPE samples. So, you know, this is sort of the worst case scenario, and yet we're still able to find some of these types of variants that are really deemed to be causal with some of the different types of studies that we're undertaking here. Shifting gears to RNA, one of the more recent introductions from a product standpoint is what we call transcriptome capture. So what this does is basically takes RNA, does a cDNA prep, a complementary DNA prep from that, and then what we do is actually expose that to a hybridization event, or like an exome pull down, where basically what we're going to do is select the bases that are known transcripts, and we actually use the same bait design that we do for our exome, but then we're able to do sort of differential expression analysis based off these data. And this works very well with relatively low RIN scores, and again, we're using the DV200 metric as a cutoff up front of this, but it's really useful coming out of FFPE, because now we can take FFPE and we can look at both exome data, and then orthogonally look at the RNA sequencing data that's associated with that, and I'll show you an example of that in just a moment. One of the things that we were happy is that we can actually detect some fusion transcripts with this, and actually we do that as well as a typical poly-A selection. So, this is comparing this Transcriptome Capture protocol to a standard TruSeq poly-A selection, where we're using an oligo DT approach to pull out the mRNA transcripts instead of enriching for them with this selection event. And what we see here is that we're able to get relatively similar or better fusion detection with this protocol as we would with a more standard RNA sequencing approach, even if we're coming out of FFPE. And here's a great example of the utility of this. So this is something that was published last month, and basically what we did is we took FFPE tissue and looked at that, and the goal of this was to map sort of therapeutic resistance in colorectal cancer. But basically what we did is had a sample from pre-therapy, post the first drug 1, but pre-drug 2, and then post-drug 2. So we're able to map sort of the exome variants that you see on the left there, where we can, all of a sudden in the post-drug 2, we start to see a KRAS mutation arise. And sure enough, when we go on the right there and look at that, we're able to see that with the Transcriptome Capture protocol, we're able to look at overexpression of KRAS post-drug. And so we're able to look at, mutation variants and then differential expression to understand that, there's a mutation happening on the DNA level, but, sure enough, that actually is impacting the transcription, and we're able to sort of, understand that and use that to map some of these resistance mechanisms. So moving on to some of the analytical approaches that we'll employ here to really deal with the data once it comes off of the sequencer, there's a number of different strategies that we've employed and to sort of overcome some of the inherent challenges that might result from having lower quality samples as the input to the process. One of those things is really understanding and resolving those subtle artifacts. So when we talk about some of those oxidation events, our variant calling tool, Mutect, we've actually built some of the filters for the different types of deamination-induced artifacts. So when you're looking at those two plots, the yellow is really where we're looking at the specific C to T deamination artifact. And what we see is that, post-filtering, and just this is a bit of an eye chart, so I apologize. On the left, that y-axis is the top of the y-axis is at 12, whereas on the right, that y-axis is at 4. So, what we see is that we're able to sort of vastly reduce the number of false positives that are resulting from these artifacts. And again, this is a function of knowing what to look for, building the right filters upstream, and then incorporating that into your variant caller, so you're not calling these artifacts as actual positives. Another great example of using some of our informatic tools to overcome this is actually something that we did the end of last year. So we were involved in a large clinical research project, and the goal was to identify some variants in tumor normal pairs. And this was specifically a blood disorder, so we were using bone marrow aspirate as the tumor, but the normals were blood. And we started to run some pilot samples, and what we realized was some of the folks looking at the data said we actually are seeing the tumor in the blood normal, which isn't unexpected given the fact that it was actually a blood ailment. So one of the things that we benefit from here is having a relatively large computational biology and tool developers. So we have a lot of these tools that are kind of in our research space, wouldn't necessarily be applied in our CLIA lab, but what we were able to do is actually look at a specific tool, and the specific tool is called Deaton. The tin is for tumor and normal. And what that actually is able to do is basically look at the reads and be able to look at the allele fraction that's associated with some of those and be able to actually understand the level of tumor that's present in the normal. And what we were able to do is get this validated in our clinical lab in time for this project. The impact that had is we're able to do some correction, but we actually didn't use it for that purpose. We used it to actually filter the samples to make sure that the sample pairs, because this was a clinical project and had some implications, we wanted to make sure that the samples that we chose for that project actually were giving us the best quality somatic variants at the end of the day. So, by using that as a filter to decide which samples we include, we're able to draw better statistical conclusions out of the data that was produced. So this is, again, just another example of applying some filters that we've developed here to be able to identify some of these things that could otherwise cause complications downstream. Another way that we're able to overcome this is just by some of the experience that we have here. So one of the things that we do here is a concept called joint variant calling, and this is something that's a function of the GATK haplotype caller. So really what we're able to do here is overcompensate for areas where we might have lower quality or lower coverage in certain regions based on intrinsic qualities of the nucleic acid. What we do is we actually will look at when we're actually processing these in our pipeline, what we do is actually per site variant calls, and what that allows us to do is differentiate between a true homozygous reference and what we would otherwise call as potentially a false positive because we had lower coverage, or a false negative, excuse me, because we had low coverage. And basically by looking at other data that we have available that might be within a single cohort, it might be across sample cohorts, we're able to use evidence from those and sort of boost our confidence and being able to reduce these overall false positive rates. So this is something that we can do, again, within a sample cohort to look at evidence of mutations and use that to statistically understand whether what we're seeing is actually a real variant or is something that is a function of not having enough coverage in a specific region that could be the result of having lower quality samples. So just to get into some overall results, to wrap up a little bit. So we've run, over the last 5 years, we've run over 5,600 FFP-derived samples, and that number has actually increased since the slide was put together. You can see we've done that across a variety of different downstream applications, and we're able to see a very high level of success in terms of meeting deliverables, and those deliverables will vary based on the actual protocol, but those are things that we specify up front in terms of meeting a specific percentage of targets for exome sequencing at a given coverage. And we're able to actually achieve that at a relatively high success rate despite the fact that we're working with, in some cases, very low quality samples. There's a publication that I would recommend pointing to for further reading. This was published last year, so this is where we're looking at some of the tools that we've employed here for whole exome sequencing and clinical interpretation of FFP tumor samples to guide precision cancer medicine. And what you can see on the bottom there is that, really, when we're looking at some of these quality metrics that we use to gauge the success of sequencing, what you see in the orange and the red are FFP-derived. What you see in the other colors are, some of the other sample types that we would typically use as input. And what you can see there is there's really no correlation between the FFP. It's really more a function of the other things that are happening and might actually rise in lower quality sequence data. So I think by putting all of these things together, we're able to overcome some of the inherent challenges from dealing with lower quality samples and generate data that we can actually draw some logical conclusions from. So in summary, some of the strategies that we've employed to overcome sample quality. We talked about nucleic acid extraction technology, some of the automation that we've employed there, as well as optimization of some of those processes. The sample QC, so being able to look at our samples, measure the intrinsic qualities, use that as a cutoff and a predictor of downstream success. Some of the different sample preparation strategies that we've employed to overcome low quality samples, and then some of the analytical approaches which we're using to sort of filter out some of the issues that we deal with on the data analysis side. So with that, I'm happy to answer any questions, and thank you for your attention.
Thank you for that informative presentation. Before we get started on the question and answer session, I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is, can the chemogen, which uses a magnetic bead technology, be used to improve this protocol?
Yeah, there's a few advantages to using the chemogen technology, and I think it would depend on the specific protocol that was being used there. One of the advantages to the chemogen technology is that we're able to deal with relatively high volumes of blood, so that wouldn't be a situation where it would be low-quality sample, but because of the sort of intrinsic nature of adding the beads to the primary sample and then using the electromagnetic rods to be able to actually separate out nucleic acid from the other cells that are present within that, there are some advantages there. The other advantage, as I mentioned, is by using this magnetic rods as a means to separate magnetic beads from solution instead of pipette tips, you're not introducing any additional shear forces. Where really, if you take a 40 kb DNA fragment and pipette it up and down a few times, you're going to induce some shearing there, and will actually result in lower molecular weight nucleic acid as a result of just pipetting that and the shear forces that are created from that.
Okay, looks like we have time for one more question. Can this sample protocol be used to analyze copy number variation in non-exome regions?
So I guess it would depend on the specific sample preparation protocol. Certainly, I think any advantage that you're going to get from using some of the strategies that we've employed there are only going to help your ability to call copy number aberrations. Exome sequencing, as I mentioned, is particularly challenging in the sense because you're only targeting a subset of the genome in general, and some of the ability to discern some of those copy number variants is really a function of the data that you're putting into it. So, if you're only targeting 1% of the genome and there's only a certain percentage of specific, if you're, I typically separate this based off of, copy number detection, so the discovery of novel copy number variants versus identification of copy number variants. So it would be easy to target some of these known copy number variants. What becomes difficult with exome data is the discovery of novel copy number variants. So actually we're working on some tools here and looking to employ those, for production that will actually allow us to do a better job of calling copy number variants from exome data. But certainly all of the strategies that were discussed here today would only help to improve your ability to detect those.
Great. I would like to thank our sponsor, PerkinElmer, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through November 13th, 2015. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.